Today we are going to talk about the chapter 4 of our textbook, and the focus will be on the rate of nucleotide substitutions. In previous weeks, we have discussed how to calculate the substitution rate based on given sequences. So we start with the one sequence with the change to a different one. Then we talk about if we align two sequences, what will be the consequence. And last week we end up to the point we discussed how to align the sequence and how to judge which alignment is better than the other. Although there's somewhat personal ideas, but there's general. If the sequence are very similar, then everybody would do have the very similar alignment. But today we want to add another layer of differences. Talk about the rate of substitution rate. And when we say the rate of substitution rate, actually in the next slide will show you is considered about the time. So just to define the nucleotide substitution rate of nucleotide substitution rate, and we add one more parameter here is the little r. So we name the r as the rate of substitution rate, and we already know how to calculate the k, right? From chapter 3, we learn how to calculate k. That's the substitution rate from the given sequences. But now we want to calculate per site per year. So that's the definition is on the slides. So just remind you about the sequence divergence. If we compare two sequences, I'll put A and B here. Then the time on each lineage is t. So there's a 2t. We already seen this diagram before. Just remind you, the two sequences evolve independently. So the total time, the total divergence time from here to here is the time t, right? But when we do the calculation, we have to divide it by 2t because each lineage evolves for time of t. In both lineage, there's 2t of time. So that is when we talk about the substitution rate per site per year, and that's the time frame we need to put into. So this is a diagram to remind you, although you've seen that before. Then another thing we also talk about, actually we emphasized a lot last week, is the synonymous versus non-synonymous. And actually we'll talk more today about that. Then after the spring break, we'll talk about the signature of selection at the molecular level, and we'll continue on this subject because in a lot of analysis, we use coding sequence for our work, and we have huge amount of data on this. In comparison, for non-coded sequence, actually traditionally we use as a reference locus, like the pseudogene we talk about, and we'll use that more in this chapter. So when we focus on the coding sequence, we usually separate the non-synonymous, the synonymous rate. Then we also would like to compare the rate on both type of substitution. As you already know, the rate of the both type of substitutions are differ quite a lot. And that is also reasonable to for the pattern we observed because there's functional constraint on each protein. So a lot of time the sequence are quite diverged, but at the DNA level, but at the protein level, they are still quite similar. So just to give you an example, you can look at the chapters, I think it's a little bit later on page 104. And that is the one to show you the histone 4 protein. Histone, we know, is a very conserved protein. It's part of the chromosome protein in our cells. And if we compare the histones from human and wheat, you will see a lot of changes occurred at the DNA level. But as I said, it's a very conserved protein. So in that page, you also see if we look at the amino acid sequence, there's only 2 amino acid replacement between wheat and human. So this is just an example to show you there are so many changes at the DNA sequence level, but most of them are the synonymous changes, synonymous substitutions, and only a few are non-synonymous substitutions. Then we'll talk more about the protein sequence differences, but I think most of us have some idea from my previous lecture. So in a lot of cases when we compare sequences, why we like to look at coding? Because we know the property, we understand protein much better. And in the past century, that means like 10 years ago, we don't really know that many non-coding genes. We know more about our proteins working in the cell, and we know more about the functions of the protein. So that's why a lot of the molecular evolution study were concentrated at the protein coding genes. But in the past 5 years, things have been changed quite a lot, but the framework still the same. So you'll see people now can compare genome-wide differences, but we still like to separate the coding sequence and non-coding sequence as we do in this chapter. Another thing we also did start from, we talk about the matrix, the model. We mentioned 1 parameter and 2 parameter models when we want to calculate substitution rate. So this is something you have to know. And you know, with more studies, the 2 parameter models are not exactly covered all the differences we observe. So therefore, people or scientists have proposed more parameter models. So that's all covered in chapter 3. But when we look at transition and transversion, now we can have some data here, or earlier data, to look at the transition, transversion, substitution rate. So here is the typical table. You will see this type of table quite a few today, because we want to ask at different position of the coding sequence, and what's the transition and transversion rate in general. And of course, this is like in your textbook is like what they can have or what the people have done at that time. Now we have more data, but the data seems consistent with previous observation. So if we separate 4-4 degenerate site and 2-4 degenerate site, that is what we talked last week when we want to calculate K and Ks differences. So we can look at each position, including the non-degenerate site, then what's the transition and transversion rate. So in this way, we can compare the rate differences in this type. So for example, in this table, the non-degenerate sites have the transition rate at 0.4 and transversion rate at 0.38. So you can easily see there's very little differences between the two numbers. However, if you look at the 2-4 degenerate site, you see dramatic differences. Now, at the transition, you observe, or we observe 1.86. Then transversion rate is only 0.38. Okay, without any statistic, you already know the two numbers are quite different from the number we observe at the non-degenerate site. So the difference is what we talk about how Lee in 1993 developed a method use transition transversion rate to calculate the synonymous substitution and non-synonymous substitution. So just by looking at a block in the codon table you immediately found out the transitions in some codon. So then at the 2,4 degenerate site, I should specify. At 2,4 degenerate site, most changes for transition, it didn't change the amino acid sequences. But for transversion, it changes amino acid sequence. So if traditionally and also most evidence show us, if the amino acid changes occur in the protein, it's more likely to be subject to selection, right? Or so that's why. Of course, it could be neutral. So that's why I say why it's more frequent to be observed. It's not necessary to. So in the beginning of the chapter, actually, I didn't really say everything, but in the first page of this chapter, talk to you about the substitution rate we observe or we are going to investigate. Actually, it's the result of several components. So we do consider mutation rate. We talk about mutation rate could be different, and we know in different organisms, the mutation rate, the spontaneous mutation rate could be different. So this is from your genetics. In your genetics, it will tell you classically how we estimate mutation rate, and tell you the mutation rate, the spontaneous mutation rate is relatively low and in most organisms, but it varies from one organism to another. Another thing is the fixation rate. I think I have a slide coming after it. Like once a mutation occurs, in chapter 2, we talk about a new mutation occur at the frequency 1/2n. So the second is like how much time will it cost to fixation? If it's a neutral mutation, you know, or if the mutation going to be fixed, will take 4n generations time to be fixed in the population. But a gene or a mutation could be fixed driven by natural selection, by positive selection. So all these components will contribute to the pattern we observe. So of course we know transitions, the first answer was like transitions much easier to happen than transversion, so which is also true. But why it's at 2,4 degenerate sites? Because at 2,4 degenerate sites, we observe the transitions are most of the time synonymous, but transversions is non-synonymous. So without changing amino acid, have better chance to be preserved in the sequences. So that's why we observe dramatic difference between 2,4 degenerate sites. Okay. Then the second question, I hope we can get an answer. But why at the 4,4 degenerate site they are all synonymous? Why it's different? So 2.24 versus 1.47. So at least you should be able to answer like the transition happen more frequently transversion, right? So by just looking at the mutation rate. Of course, later we'll think about other components. So I tried to pick up probably the most obvious answer for the differences, but it doesn't mean that's the only answer. But it's like the most obvious one should come out in our mind is the differences in transition. Then come back say, okay, why there's no differences in non-degenerate site? Then come back to the first one. If given the mutation rate is different, right? So that is how we explain the 4,4 degenerate site. Then why the non-degenerate sites are quite similar? I think the major player at this point is like how much of the mutation can be seen or can be observed in our sequence data. Remember, mutation could randomly happen at any position. at any time. Yeah, although it's very low, but it can happen at any site. But not every mutation can raise to a high frequency or even to a fixation be observed by us. So when we sample sequence from the population or from several different species, those are the sequences, like probably it's a majority sequence in this population. Then for each mutation, they are not all the like a very rare mutation. It is most of the time they are at certain frequency. So we have the chance to observe them. If a mutation is very deleterious, then it occurs, but immediately will be wiped out by natural selection. So that's what we talk about the negative selection, right? So those are very bad mutations. They will immediately disappear in the population. So all the substitutions we observed here are the ones which can still preserve in the organism, so that organism still can reproduce. Then that means those amino acid differences cannot be too bad to the organism, to the individual. So that is like a strongly biased by the selection pressure, by the functional constraint at the down-degenerate site. So we roughly, so if I say compare these three types of differences, one is primarily dominant by selection, functional constraint. When I say selection, doesn't mean the selection occurs in any places, but the observation we have here is pretty much shaped by natural selection, positive and negative. And the 2,4 degenerate site is thinking about when it's occurred, whether it changed amino acid sequences. So the one without changing amino acid sequence have better chance to be preserved in the population, because we can see it's close to a neutral mutation. Most of them could be neutral because they didn't change the amino acid. But the one changed to amino acid will be similar to the non-degenerate site, right? And actually, you also observe the rate of substitution is similar. So the 0.38 actually exactly the same as the non-degenerate site for transversion. Then for 4,4 degenerate site, most of the differences contribute by mutation differences between transition and transversions. Okay, so this is something I would like you to be able to have the ability to diagnostic the data. So I spent like 15 minutes on this single table. I don't want to just show you, oh, you see transition, transversion are different. Actually, in this chapter, everything is like that. Transition and transversion are different, and different regions like exon, intron are different. Coding sequence and pseudogene are different functional constraints. So everything is like that. So we try to do what this chapter to do is tell you we can estimate the rate, and we are interested in the rate differences between genes or among different regions of the genome. And we want to know why. Why is the reason? And that's how we practically doing the molecular evolution research. And we like to use the data to estimate the divergence time. So very quickly, Professor Hu will talk about the molecular phylogeny. And we like to know the species A diverge at certain time. And we want to compare that with all the other knowledge we had in the past in biology. So those are the things we are interested. So I've spent this like much time in this single table. I would like you to have the ability to ask yourself, once you get results from your own data or in our laboratory course, you will do a little bit of calculation. For each section, you will get numbers. Like you can calculate genetic distance, So we get the distance data. So you know A and B differ by how many differences. But what does that mean? And this is another example. We observe the rate differences between transition and transversion, which we already know. But what does that mean? And why sometimes they are very similar when we dissect into different categories? So that is a lot of times we are doing in the laboratory. Try to look at the number and try to figure out what's the underlying mechanism shaping the evolution. So this is the practice. And this one we already seen for the first chapter, just remind you a gene structure. And we also talk about pseudogene. Pseudogene is a very popular topic in molecular evolution since very early on. And we already talked about the definition of the pseudogene. And in this chapter, we want to show you a very early study compare, at that time, a large number of genes and try to understand the rate differences. So when I say the rate differences, is the differences in the substitution rates for different regions of a gene.